Okay, welcome everybody. My name is Mark Gardner from AMG, and we're going to kick off the 6th, I think it is now, Global Health Compound Design meeting. And I'd like to say a big thank you to Anna Gaulton from EMBL-EBI and Jérémy Besnard from ExScientia, who are going to talk to us about ChEMBL. Just whilst I'm introducing things, I want to launch a quick poll question just so that we have an idea of your experience with ChEMBL and also with matched molecular pairs, which Jérémy will talk about. So if you could just take a look at the poll and click the answer that's most appropriate for you, that would be great. So just a couple of other housekeeping things. Please feel free to use the question panel to communicate with us. There's a question tab there, and those questions will be recorded, and if we don't get around to them in the meeting live, we can address them afterwards. If you do want to speak, you'll need to raise your hand because you'll be in mute until both I unmute you, and if you've muted yourself, you'd have to unmute yourself as well. So We'll get started in just a minute. So I can see that about half of you have voted so far. So if there's any more people who haven't made up their minds, rather a lot to read, I apologise. We will kick off, and Anna's going to talk about ChEMBL itself and start off, and then Jérémy will take over with a couple of use cases from his own work. So I will now close the poll. Any last minute votes? I guess some people may be just joining us. You can have a quick look at the poll. Okay, I'll close it now and I'll show everybody. So hopefully you can see that yeah, about a third of people have used ChEMBL and know what matched molecular pairs are. And about a quarter haven't used ChEMBL and are not familiar with matched molecular pairs. Fantastic. So I will now turn the presentation over to Anna and let Anna do her presentation. Thank you, Anna. I think I may have to hide the poll results. Okay, there you go. So I think, Anna, you should be able to make your screen live now.
Okay.
That's it.
You see that fine?
Perfect.
Okay, thank you, Mark, for the introduction. So I work here at the European Bioinformatics Institute on the ChEMBL database. So I'm just going to tell you a little bit about ChEMBL and how this can be used for drug discovery and drug design today. So just to give you a basic overview of the aim of ChEMBL. And primarily what we do is we take information from the published scientific literature, so things like papers from Journal of Medicinal Chemistry, for example, and we extract information about the compounds that have been synthesized, the assays that have been performed, the bioactivity data from those papers, put it into a structured, organized form that people can search more easily, and allow them hopefully to derive insights and tools and resources for drug discovery from that data. So we try to track the whole drug discovery process through from very early target discovery through lead optimization, clinical stages, hopefully to final approval of drugs. And the medicinal chemistry literature kind of tracks the early phases of drug discovery, and then we also collate information about clinical candidates and approved drugs, and that can span the development phases. So in addition to ChEMBL, which I'm going to talk about today, we also have a more recent resource called ChEMBL, which is a patent database, and that includes information about compounds even earlier before they usually get published in medicinal chemistry literature. So we really want to follow the whole drug discovery process through. And a little bit more detail about the data that we actually extract. So we go through full text articles in primarily medicinal chemistry journals, so JMedChem, BioOrg, MedChem Letters, and things like this. and we manually extract information about the compounds that have been synthesized, including their full chemical structures, the assays that have been performed on those compounds. We map that assay information through to targets, which could be molecular targets like proteins, or could be things like cell lines or organisms. We extract all of the quantitative bioactivity data from those publications. So this is not just binding data such as KIs, this is also data from functional assays and also ADME and toxicity measurements. We put this all into structured form so that people can search by target, by compound, pull back the types of data that they're interested in. And the most recent release of ChEMBL, ChEMBL 21, this just gives an overview of some of the statistics, the different data sources that we capture within ChEMBL. So at the top, I said the majority of our data comes from the scientific literature, and this is the majority of the assays within the database, and around 5 million activity data points. But we also include information from different databases and projects. So OpenTG Gates and Drug Matrix are two kind of toxicity projects that made lots of data available, so we put those into ChEMBL. We also exchange data with PubChem bioassay database, so we have information in ChEMBL from PubChem. And then more recently, we have a lot of information from deposited data sets. So particularly in the area of neglected diseases, we have Medicines for Malaria Venture, Drugs for Neglected Diseases Initiative, who regularly deposit data in ChEMBL, and a lot of other collaborators as well who deposit data. And then we also have some interest from pharmaceutical companies in starting to deposit data within ChEMBL as well. So AstraZeneca deposited a large set of PK data that they were able to make available. We have a number of supplementary bioactivity data sets that other pharmaceutical companies have made available. And these are things typically where they've published information in the literature, but they have supplementary data which they never published. And so this is an increasingly interesting area for us. We welcome people to deposit their data directly with us, as well as us collecting it from the scientific literature. We do a lot of curation on the data that we extract. So we have an automated process for standardizing the compounds to represent them in a particular way. We strip salts from compounds, so we register the particular salt form that was used in the assay, but we also generate the parent compound so that people can collate data across different salt forms with the same compound. We calculate a wide range of different physical-chemical properties of the compounds as well and store those in the database. And we have manual curation of chemical structures as well. So if we spot a structure that's been drawn incorrectly, could be missing stereochemistry, it could have bad valence or something like that, we will go in and correct those compounds. We do this both on new data sets, but also we go back over the historical data. If somebody spots a problem, we'll go in and fix those compounds. We also do a lot of data standardization on the actual activity measurements that we extract from the papers. So people have lots of different ways of saying things like IC50. So we record the actual published types that were mentioned in the publication, but we also standardize these to kind of standardized activity types. So, you know, example on the top right of all the different ways people might say an IC50. And then below this, all the different units that they might specify. And this is just a subset of all the actual different measures of molarity that we do see within ChEMBL. We try to convert all of these different types of molarity measurement into nanomolar units so people can easily compare data between different assays. We also try to flag data that could be incorrect. So we look at the units of the assays. So if you have, for example, an IC50 measurement with units of milliliters or micrograms, we'll flag this as being a non-standard unit for that activity type, so it's probably incorrect. But we need to go back to the original publications to work out if it's actually the units or the activity type that's incorrect quite often. Similarly, if the IC50 is abnormally high or low, we'll flag this as being outside of the typical range for that measurement type. So if you're interested in a particular target and you want to find as much data as possible, you might want to include these types of measurements and then go back to the original publications. But if you're doing high throughput data mining and you just want a large clean data set for modeling, for example, it'd be wise to exclude these measurements that have a data validity comment flagged to them. The other thing we do is we try to calculate what we call a p-ChEMBL value. So for dose response type measurements, things like IC50s, KIs, EC50s. We calculate a negative log value for those so that you can easily plot these on a graph, for example. And then finally, we try to flag potential duplicates within the data. So you can see this example at the bottom. There are 2 rows within the ChEMBL database which have the exact same compound, the same target, the same activity type, the exact same nanomolar measurement, and they're from different papers published 2 years apart. It's highly likely that the second measurement is just a repeat citation of the earlier value, and it's not actually an independent assay measurement. And so again, we flag these instances so that people could exclude them if they're doing modelling and they don't want to bias their data set. I mentioned that we don't just collate binding activity data. We have a wide range of functional and ADME measurement types. So the actual in vitro binding assays make up only around 18% of the total bioactivity data within ChEMBL. There's a huge amount of data from cell-based assays, tissue-based assays, and even in vivo assays as well. So we really want people to be able to search and navigate all of these different valuable measurement types. So we have tried to classify the assays within ChEMBL and map them to a wide range of different ontologies so that people can perform more sophisticated queries of this data. So rather than querying just by a compound or a target, you can ask more sophisticated questions, such as get all the compounds that have been tested in a toxicity assay in HepG2 cells, for example. We use the bioassay ontology to classify things like the assay format, so whether it's a cell-based assay, tissue-based assay, an in vivo assay. We also classify the endpoint types, so we use the bioassay ontology terms for things like IC50s, KIs. We have assay types that we assign each assay a type of either binding, functional, toxicity, or ADME measurements, so you can group the assays that way. We also classify the protein targets according to their protein families, so if you want to retrieve all assays for GPCRs or kinases, for example. And then finally, for cell-based assays, we map these to cell line ontologies, so you can query by the particular cell type that was used. We're also doing this for tissue-based assays in the next release of ChEMBL as well, and mapping those to tissue ontologies. And then the other thing that we do is to try to catalogue data around approved drugs and clinical candidates. So we take all the new drug approvals from the FDA Orange Book and we enter those into ChEMBL. We're also using clinicaltrials.gov to try to identify clinical candidates. And we also track USAN applications. So people typically apply for a USAN name for their compound somewhere around the kind of mid-clinical stages. So this is a good way to also track compounds that are entering development. And we do this for just small molecules, but also for biotherapeutics. We're not just restricting to small molecules. And we have these little drug icons that we assign to all of these compounds. And these give an idea, on the left-hand side, the green icons give an idea of the type of compound structure. So whether it's a small molecule, a natural product, a monoclonal antibody. We also assign whether it's Rule of 5 compliant, whether it's the first-in-class compound. And then for approved drugs that have products on the market, we also have these blue icons which show whether the drug is dosed orally, parenterally, topically, whether it has a black box warning associated with it, whether it's available over the counter or prescription only. So if you want more information about the drugs, these icons are a really nice way to get that. And then for the FDA-approved drugs, and also now more recently for the clinical candidates, we try to assign the efficacy targets. We do this manually. So what we mean by efficacy target is a target with which the drug directly interacts and that's responsible for the efficacy in the approved indication. So we're not really considering targets that are responsible for adverse effects, and we're not assigning new targets purely on the basis of pharmacology data. We look through things like prescribing information, published scientific literature, and we really need to find evidence that that target is related to the indication that the drug is used for. And we also assign things like the action type, so whether the molecule is an agonist, an antagonist, whether target's something like a protein complex. We try to assign the subunit to which the drug binds, where we have that information. And we try to deal with the targets of the drugs in a robust way. So Obviously, people generally think of drug targets as being proteins, and in the vast majority of cases this is true, but there are also drugs that bind to other molecular entities such as DNA, ribosomes, and even small molecules, so there are antidotes for things like methotrexate, for example. But even within the protein targets, these can be subdivided into single proteins, something like phosphodiesterase 5, or protein complexes, where the drug actually binds to a protein complex consisting of multiple subunits. There are cases where we know the target is a protein complex, so for example GABA-A receptors, but we don't know the exact subunit combination that is present in that target, so we call these a protein complex group. It's sort of a fuzzy complex. And then there are also drugs that are non-specific, that bind to all members of a closely related family, such as muscarinic receptors, so we call these a protein family. And there are a few cases of drugs that also inhibit protein-protein interactions as well. So we really try to represent the targets in a robust way. And for things like protein complexes, we then annotate the subunits which we know the drug interacts with, where we have that information. So that was all I really wanted to cover around the data in ChEMBL. And I'm just going to talk through a few of the general applications that people can use this data for, particularly with respect to drug design. So there are obviously simple applications, so if somebody's interested in a particular compound, they can use the bioactivity data within ChEMBL to try to identify targets that that compound might bind to. And similarly, if you've got a target of interest, you could use ChEMBL for identifying chemical tools that might be available for that target. But because it is such a large, diverse data set, we can also try to use this for more systematic data mining approaches and try to come up with predictions based on the data. So for example, if we take all the approved drugs and medicinal chemistry compounds and their targets, can we try to derive rules for drug discovery success or failure? So what features make a successful drug target? This is quite a complicated question to answer, but perhaps a slightly simpler form of that question would be what features make a protein druggable by small molecules? Or what features of a compound contribute to good oral bioavailability? And similarly, you could ask questions like what chemical groups may be associated with toxicity if you've got enough toxicity data already. So just a brief overview of the kinds of things that we can do around druggability. So if you have a catalogue of all the approved drugs and their targets, you can try to derive models that will predict whether other targets might be druggable. So one way of doing this is a structure-based approach. So something we've previously done within our group is to take all the PDB structures for drug targets with their ligands bound, look at the properties of the cavities that those ligands bind in, and try to train a predictive model to see what properties indicate that a cavity could bind a drug-like small molecule. But similarly, you can do this on a more ligand-based approach, so use the ChEMBL data to say which targets have small molecules that are known to bind to them. Maybe then other members of those protein families might also be druggable. And you can build various different types of models and rules using this data. One of the first things that was done when ChEMBL came to the EBI was to use it to annotate the newly sequenced genome of schistosoma. John and some of his colleagues basically used the ChEMBL data to, for all the genes in the schistosoma genome, to try to identify those that were homologous to known drug targets or had compounds available within ChEMBL. And this is a good way to prioritize novel genome sequences to try to identify quick wins in terms of targets that could be potentially druggable. You can also do this in other ways. So I think Jérémy is going to talk a little bit later about QED, which is a measure of drug likeness. So I won't talk too much about that. But if you have a measure of drug likeness for compounds, you can then apply that to the targets. So you can rank a set of targets according to how drug-like their ligands might be. Another application of the ChEMBL data is to build target prediction models. So if we have a lot of active compounds within ChEMBL for a particular target, we can build a kind of machine learning model for that target, or QSAR model, to try to predict other compounds that might be active against that target. There are a wide range of different methods that people have used for this. Within the ChEMBL group, we've used these multi-category naive Bayesian classifiers. to do this, and these models are available if people want to use them. There are also lots of other approaches. For example, the C algorithm, which has been developed, just uses basic chemical similarity between ligand sets. And there are other methods that use 3D similarity or combinations of protein and ligand descriptors. And there are lots of open source tools that are being more and more available to build these models. So the models that we build within the group, we use scikit-learn and RDKit. And we have these IPython notebooks available, which you can use to kind of walk you through the process of how to build and use these models. Again, a couple of examples here from the literature. So the paper on the left is from Grace, who works in our group as a postdoc, and she's used target prediction models based on ChEMBL data to try to identify the targets for compounds that were identified as being active in the tuberculosis screen. And she's actually identified a number of DHFR inhibitors using these models. And then the paper on the right relates to the C algorithm that I mentioned previously. So this algorithm is again trained and tested on ChEMBL data. I'm going to just briefly mention matched molecular pairs, but Jérémy will no doubt go into this in a bit more detail later on. So the concept of matched molecular pairs really involves looking for pairs of molecules within a data set that differ only in one chemical group, and then looking at the effect of that change on a property of interest. So this could be something like potency against a protein target, or it could be properties such as stability, which is what I'm showing here. So if you go through ChEMBL and you look at all the pairs of molecules that differ, in the top example, for example, these molecules differ just by the addition of the fluorine group. If you add that fluorine, you get an increase in stability. Whereas in the other two examples, in the middle and the bottom rows, the change in the functional group actually decreases the stability. So if you go through a large data set and you find that 9 times out of 10 adding a fluorine will increase stability, these are rules that could be applied to your own compounds in terms of design. And again, there is a lot of published literature around this approach. So Jürgen Bayer's group do a lot of work around matched molecular pairs and activity cliffs based on ChEMBL. And Jérémy's going to talk a little bit about the work they do around matched molecular pairs as well. So I'm not going to go into that too much here. If you want to use the ChEMBL database for your work, there are various different ways that you can access this data. So we have a simple web interface available, the URL shown at the top there. You can search the data by compound, by target. You can do structure, substructure and similarity searches of the data. You can do keyword searches, for example, pull back all the bioactivity data, and it allows you to download subsets of data as well. But if you really want to do something more high throughput data mining kind of approach, this might not be the best way to access the data. We also have a really comprehensive set of web services. So these are REST-based web services. There's an extensive documentation which allows you to test all the different services and see what parameters they require. And this is a good way if you want to do something more programmatic to access the database. And then finally, if you'd like to have ChEMBL on your laptop and everything in one place, we have a virtual machine which we call My ChEMBL, and this is a really, really powerful tool. So it includes a copy of the database, a simple web interface, a copy of the web services, but it also includes a wide range of IPython notebooks. So these are tutorials that talk you through how to do various analysis of the ChEMBL data. There's integration with KNIME, for example. So this is a really nice tool, you know, if you're going to make use of ChEMBL a lot. So I just wanted to acknowledge really the ChEMBL team. We're going to have a new group leader, Andrew Leach from GSK, is joining us in September. If you want more information about ChEMBL or any of our other resources, such as ChEMBL, which is a patent database, or UniChem, which is an identifier mapping system that links lots of chemistry databases together, you can go to the EBI website and find more information there. We also have a ChEMBL blog, which has lots of information and announcements on it, and a Twitter account. We have a ChEMBL announced mailing list, which you can sign up to, and you'll get alerts when new ChEMBL releases come out. And then there's a ChEMBL help email address, so if you do want to contact us if you have any questions or if you spot an issue with ChEMBL, you just contact us on that address and we'll get back to you. And then there are a number of papers about ChEMBL, SureChEMBL and Unichem as well that you can find more information. So I think that's all I really wanted to say for now, so if there are any questions.
That's fantastic. Thank you very much, Anna. So one question has come in from Chris. Chris, do you want to ask your question? Chris Swain, you can speak, Chris, if you are unmuted. I've unmuted you. Oh, can I ask? Sorry, no problem. Okay, any plans to better support rendering small molecules that contain metals such as cisplatin?
We don't have any immediate plans. We do kind of keep the structures for those within ChEMBL, we just don't display them anymore. They caused us a lot of problems, but it's more to do with the file format. So things like INCHI doesn't really support organometallic compounds very well. So, you know, if we find a good solution to this, then yes, we'd be happy to do it, I guess. But in the immediate term, we don't have any plans to do that.
Okay, so anybody out there who's got some good ideas on ways to do that, you'd be pleased to hear from them. Excellent. Just a note that I will, in the email follow-up to this, I'll probably include a couple of references also about accessing the ChEMBL data through Data Warrior, which quite a lot of this group have seen previously. So, you know, there'll be a little bit of extra information in the follow-up email. Okay, if there are no other questions, we'll move on to Jérémy. Thank you again, Anna.
No problem.
Hello, can you see the slides?
Yep, that's great, thank you.
Yep, okay. Good afternoon. Thank you, Mark, and thank you, Anna, for presenting ChEMBL. So actually, some of my slides will be quicker like this. So I will present some work that we did in my group, so in ExScientia now, the company, but also during my time at the University of Dundee. So we'll be on QED, how to quantify drug likeness, and also de novo design, which is more how to combine the model building and transformation you can extract from ChEMBL. I have problem with PowerPoint with all the thing going. Sorry. I have problem with my PowerPoint where all the slides move by themselves. I'll try to see if I can fix it. So, sorry, I don't know. Yes. Hopefully now it will be better. Sorry about that. So I present two topics, QED, quantitative estimate of drug likeness. So we show what it is and the extension of it, so it's not limited to just drug. And then ChEMBL as the information for de novo drug design. So what is de novo drug design? How we can extract transformation from ChEMBL and then use the activity models that Anna mentioned, and show an example on putting all of this together to find new compound. So as a quick disclaimer, all my work, I used a software called Powerplan Pilot from BioVia, so that's proprietary software. However, everything presented can be reproduced with open source software or other toolkit, and I will try to show it as much as I can with a different group doing the same. So first, QED. Our goal when we designed this function was to move away from filters and the drawbacks of filters when doing drug discovery. One problem with filters, it doesn't rank compound. You just have a set of it pass or it fails filter, and the borders can be quite limited. So as an example, if you use a rule of 5 and molecular weight of 500, a compound with a molecular weight of 499.99 will be considered good, but a compound with a molecular weight of 500.01 will be considered bad. However, they are very close. So just to show, so here are some different rules that exist. The famous rule of 5, which are quite known, but some other group defined other rules like Weber with using rotatable bond and polar surface area, and Gauss using other function. But they are all binary, true or fail. So as an example, if we have this large square, blue light square of compounds, that's our library, and our capacity to screen is a dark blue square, and we need to select compounds. If we apply filter, we may end up with actually more compound passing the filters and our screening capacity. And then we need to spend time to reduce this again. And how do we do it? We need more filters, more clustering, and it becomes a bit more manual work. Also, the possibility is after applying filters, you have less compounds that you will want to screen. And here again, may have to play with the filter. So now can you make molecular weight instead of 500, 510, and moving around just to reach the desired size. While if you had all your compounds ranked by desirability, then you can just take the top X compound to fill your screening deck. So that's the idea between QED, so quantitative estimate of drug likeness. What we want is a measure between here 0 and 1, with 0 bad and 1 being good. And 1 will be try to represent drugs. So we took from ChEMBL a set of around 800 orally drugs. So as Anna mentioned, ChEMBL extracts the information for drugs. And then all those drugs were represented with physical-chemical properties that are well known. Molecular weight, log P, number of hydrogen bond donor, acceptor, polar surface area, number of rotatable bond, aromatic ring, and a set of structure alerts which are used for lead design, and those alerts from our collaborators in Dundee University. So all those features are known and can be represented. Then how we do to get this value between 0 and 1. So here you see the 8 properties and the distribution for the 770 drugs for all of the different properties. We see distribution, and in black blue is the function that represents this distribution. And the idea is when we have the most compound, it will get a value of 1, and on the edges it will get a value of 0. So a compound with a molecular weight around 350 will have a desirability of molecular weight of 1, while if it has a molecular weight of 800, its desirability function would be almost 0. Then all of these functions, which varies between 0 and 1, can be combined into one single measure called QED. I think to show how it can be used, if we used the rule of 5. For the set of drug, we see that most of them pass or fail, but it's very binary. While using QED, we can see the wide range of compounds, some having good QED and passing Lipinski rules, but some others are failing the rules. But give more granularity than just yes or no. So here are some examples of the best drug using QED. So usually these are quite small compounds, and the second row are the least drug-like drugs, so which are usually very large compounds. But one approach we wanted to do was to avoid having a black box, and you want to be able to understand why you get a certain score. So here are 3 compounds with different QED scores and a radar plot. So on each of the lines on this plot are the 8 properties. The red line is a compound we are interested in. The dark blue line is a median from ChEMBL, and the blue shade is a ChEMBL distribution. So we get an idea where other compounds are. For example, the first compound has a very good QED, and we can see that most of the red is on the edge, so close to 1, and so almost perfect. As we go down, the second compound has a lower QED, and we can see that the edge bond acceptor is very low desirability, so that's almost close to 0 for the red line in the middle, meaning that this compound has likely too many edge bond acceptor compared to the known drugs on the market. Finally, the latest compound has even worse QED. We can see the red lines going close to 0 in multiple places, and in particular from the molecular weight, edge bond acceptor, and rotatable bond. And this compound may be a bit too big and too many bonds. So this can be used on any of your compounds that you design. It's not limited to the drugs, and you can rank compounds based on how drug-like they are. The function need to be derived first, but once you know how to calculate them, it's just a formula, and you just need the different molecular properties of a compound, and then the QED can be calculated using any software. It can be from your database, Python, or even Excel. It was mentioned by Anna, but the QED value are calculated by in all the compounds in ChEMBL, so you can actually just get the QED straight from ChEMBL. And it's not limited to the software we used. So here's an example where a company called Silicos IT, they provide a script to run QED in Python using the chemistry toolkit RDKit, and they redefined all the parameters of the different function to fit the values from RDKit. So A log P could be slightly different, so it's possible to redo it. And finally, on QED, our paper described a method to derive it from oral drugs, so it can be available via the web interface. But it's not limited to oral drugs, so you can define the same type of function and scoring for different drug sets, maybe non-oral or drugs which are not yet approved, but also on any set of compounds. You may want to derive how similar your compound would be to compounds active against a specific target or family of targets, and use it to design your future compound or rank your own internal library. I will move to my second topic, de novo design, which we used a different set of information. So what do we try to achieve with de novo design? So what we want to do is to design novel compounds, usually in silico, with a set of transformation. We generate loads of compounds, and then we use different models to reduce the set and synthesize and test our compounds. So I will use a paper we published as an example, so Anna mentioned it, but again, it's not limited to this example. So what are we doing? So we try to have an algorithm. So we take some compounds as input. We'll transform those compounds using a set of transformation into a very large library. It could be millions of compounds. Then we use some models to take the best compounds. We can also use QED at that stage. And we may try to do this step multiple times to have better compounds. Then we will use selection and have maybe around 10-20 compounds that we can ask chemists and biologists to actually test experimentally. So I will present the step in red, how to set transformation and how to derive models from ChEMBL. So first we need some transformation, how to modify a compound with a computer. So we try to reproduce what a medicinal chemist would do. The idea at the end is to make a compound, so we don't want something too crazy. And how to extract them from ChEMBL, the source will be matched molecular pairs. So we'll come back to it on the next slide. Or if somebody has more time, can do a manual work and look at pairs of similar compounds within a paper. So ChEMBL can be extracted by article and then draw manually how to go from one compound to another. But the matched molecular pairs will make it automatically. So briefly, so the matched molecular pair, say it's a single transformation between two compounds. So for example, here, from the ethoxy to the methyl carbamate, and we see that it's a single cut between the two compounds, and we can encode this transformation as drawn on the right. And this transformation can then be applied to any new compound to transform any ethoxy into methyl carbamate. As Anna mentioned, we can gather information around this transformation on activity, solubility, log P, and then transformation can be selected on which one want to apply to generate new compounds. If you want to do it, an easy and fast way is to actually group compounds per article in ChEMBL and then generate MMPs, and you will get also the direct information around the activity using the same assay. And just to show that MMPs are quite widespread and used in different places, here are different software you can use them. So KNIME, which I think was mentioned in the previous webinar, use the RD kit to generate matched molecular pairs, but also other software like OpenEye. And finally, you can look at them on the Swiss Bioregister database. So they have a website where they extracted every MMPs from ChEMBL, and one person can search for transformations that can be used on a specific compound. So now we can generate compound, but we need to be able to rank them to be tested, and the most important would be to have an activity model. So we don't want to synthesize and make compound randomly. We try to have compounds which have most chance to being active in an assay and to try to reduce the amount of resources we spent on the project. We use the same models as in ChEMBL team, the categorical model. It's quite simple to define because it's just active or inactive, and a threshold can be changed depending on your need. In here we take 10 micromolar as an example of active compound, and inactive compound can be used of all the rest of ChEMBL. with the idea that if a person made a compound for a certain target, it's likely to be inactive against the others. One advantage to use categorical model is the resistance to noise, and you can merge assay between different papers without worrying too much. So for example, if a compound was tested and got 100 nanomolar in one paper but 600 nanomolar in another paper, they will be both considered active and won't affect the model. While if you try to have a very precise model to predict the exact activity, this can be a problem. So how do we merge all of it together? So in this example, we will try to define a new ligand for a dopamine D4 receptor, which goes in the brain, so it goes a good CNS profile. What do? We take all the data from ChEMBL and we annotate if it's active against D4. So we can use the p-ChEMBL value or just assay, and we say if it's below 10 micromolar, you are active, and any of the compound from ChEMBL is inactive for the model. And we need some compound to transform. So what we did was to take all the drugs and use a multi-categorical model and see if we could predict a target which was not known for the particular drug. And we found that this drug called donepezil, it's an acetylcholinesterase inhibitor, scored very well for our dopamine D4 model, but there was no activity reported for it. We confirmed the activity and we decided to use this as a starting point and improve that side activity. So here is an illustration. So on the top left we have donepezil. It's colored by the model score. Orange means that the model likes this part of your compound. It's very good for D4. Blue, the model doesn't like that part, and gray is neutral. And what we try to do is to transform this compound, multiple steps, and then score until we have a compound with a very good score for D4 and also Novo. So the fourth compound was tested and was selective and active, but was not Novo. So then we used the same algorithm and the same transformation to generate new compounds. We can see at the end that some of the compounds have blue atoms, which are actually not very good for D4, but those were introduced for the selectivity. And so we had other models for different GPCR were used inside this to actually create a selective compound, even if it means a less good primary activity. The final compound, printed here, was novel, potent, crossed the brain barrier and showed in vivo activity in mouse. And all this work here was done just using public information. So models were built from ChEMBL, transformation were derived from ChEMBL. And the only steps were few intermediate were made and tested to see if we were on track. As mentioned, all my models were built in pipeline pilot, but they are 99% the same as the ones that Anna mentioned and that were built and available online. And they are the one where if you go on ChEMBL interface, you can see some target prediction for some of the drug, including donepezil, and D4 is one of the predicted target. So as a conclusion, we can see that using public data from ChEMBL, or if you have internal combined with it, and different software, it can be proprietary, open source, it's possible to derive properties, so like QED, which try to represent in an interpretable way a set of compounds. It can be drugs oral drugs, topical drugs, or just active in an assay, can be phenotypic assay or binding assay, and can use to be scoring new molecules, but also scoring your screening deck and which compound do you want to test in your assay. We can also build transformation sets and a range of activity models, could be for your primary activity or your anti-target, that can be used in combination to explore new chemical space and try to help you on making your next compound. So I want to thank so from the ExScientia / University of Dundee, so the work is combined. So professor, but also most of the QED work was done by Richard Bickerton. So this is work, and also the ChEMBL teams that helped via the help email throughout the last 7 years on information about the assays and input. And thank you all for your attention.
That's great, Jeremy. Thank you very much indeed. If anybody has any questions, please submit them on the question panel or raise your hand if you'd like to ask them yourself. Chris Swain has a question. Have you tried using ChEMBL to check for patent novelty?
Yes, but at the moment we used it only for identity check. But yes, so we download ChEMBL, we mirror it and then do a check for novelty that way.
Okay, great, thank you. Feel free to continue to raise your hands or submit further questions. I've got a couple of concluding remarks, but also just before while I make those and talk about future meetings and things, just for a bit of fun, I have a second and final poll for you. So you may have heard recently, we've been talking about, big data in some ways, big data in medicinal chemistry anyway, and to some extent, I guess you could say that some of what Jeremy has been talking to us about is related to artificial intelligence. So you may have heard that doctors and lawyers could soon be replaced by artificial intelligence. That was a claim made by Andrew Moore. So what about compound designers? Be interested to know this. I don't know whether this question has been asked of experts in this field before, but if it hasn't, you are the test answerers of this question. You can determine whether or not you think that's likely to happen to compound designers. So whilst you're turning your attention to that, there is another question here. No, no, that's okay. That's all the questions that came through. So just in terms of finishing in terms of the follow-up email to this meeting, you'll get a link to this, the presentations. You will also get, I'll add some information about ways to access ChEMBL in other ways and also other databases. I've had some information from the Open Facts team, for example. So we'll put some information in the email follow-up. Just see here. Okay, yeah, one more question come through. Thank you. Is it possible to access this information right after this session? The talk will be available. It'll take a little while to process and make available. It'll probably be about an hour, I think. If you mean ChEMBL and things like that, that's available online now. If you want to follow up to that answer, by all means do so, and I'll try and interpret it, or indeed raise your hand and I'll unmute you and you can ask that yourself. So in terms of future meetings, would love to hear from you if you're interested in giving presentations at future meetings. We may take a bit of a holiday in August for these meetings, but we would love to hear, in particular, case histories of drug discovery, particularly in neglected diseases, but it doesn't matter if that's already published work, because it's always good to hear people talking through their work, as Jeremy and Anna have done here today. And also we're going to get a couple of presentations on the application of predictive models, and Jeremy's talked about some of that here today, but it would be great to hear some more examples of how predictive models, we're thinking particularly of models that are predicting in vitro endpoints, have been applied. So particularly interested in the application rather than at this point much more on the detail of developing the models. So for future meetings, those are some of the areas that we're interested in, but really anybody that's got an interest in making a presentation to this group, please get in touch. So let me just close this poll and show you the results. So basically you all think you're, well, no, 60% of you think you're less replaceable than doctors and lawyers. I'm relieved to hear that. Doctors and lawyers may not be, but anyway. Okay, any final questions? Okay, I don't think so. So thanks very much indeed once again to Anna and Jeremy. It's been a really interesting session, and it's great for you to take the time out to give those presentations, guys. And thanks very much to you for attending, and you will get follow-up emails shortly. And hope to see you again at a future meeting. Thanks a lot. Bye.